Yeah, in today's class, we start with this photolysis of water results in evolution of, that is the question. Okay, this is most important question, the photolysis of water. So where we find this photolysis of water? We find this in the photosynthesis. Okay, photosynthesis is divided into light phase and dark phase, right? Light phase and dark phase. So in the light phase, again there are two photosystems, PS1 and PS2. So I will show you once. Yes, I explain you again. So in in photolysis, what happens? There are two photosystems, PS2 and PS1. In the PS2, the reaction center is called as P680. So at PS2, so in the light phase, we see two types of the cycles. One is cyclic photophosphorylation, another is non-cyclic photophosphorylation. In the cyclic photophosphorylation, we see the only one kind of the photosystem, that is PS1, photosystem 1. It only participates. In the cyclic photosystem, there is no photolysis of water. But in case of the non-cyclic photosystem or non-cyclic photophosphorylation, we see the participation of the two photosystems, that is PS2 and PS1. But only at PS2, this photolysis of water takes place. This is also called as oxygen evolving complex, OEC. Okay, in the OEC, what happens? The H2O, the lysis of the H2O, splitting of water, it is also called as splitting of water. In presence of the light, water splits into H plus ions and oxygen and electrons. These three are produced due to the splitting of water. So this splitting of water, this process only occurs at photosystem 2 in the presence of the light in the daytime. Okay, so this is very important process, because in this process oxygen is evolved. That is the oxygen we all depend on. Okay, all the organisms depend on the oxygen for the respiration. This oxygen is released from the plants in the photosynthesis. In the photosynthesis, particularly in the light phase. In the light phase, specifically and particularly, it is released at photosystem 2 during the non-cyclic photophosphorylation. The complex which is responsible for the photolysis of water or splitting of water is called as oxygen evolving complex. And for this process, two ions are very important. One is manganese, another is chloride. Okay, Mn plus 2 and Cl minus. These two ions are very important for the evolution of water from the evolution of the oxygen from the H2O during the photolysis of water. So it is very important. The electrons which are released by this process, photolysis of water, these reach the PS2. Okay, they reach the P700, that is the reaction center of the PS2. Okay, some people are joining. So next, so now you understand photolysis of water results in the evolution of oxygen. Okay, why I explained all this? Because they may ask different questions on this. What is OEC, oxygen evolving complex? What are the ions required? Manganese and chloride. Okay, along with the oxygen, H plus ions also released, electrons also released. Okay, next question is the number of chromosomes in Arabidopsis thaliana. So this I explained you earlier in Arabidopsis thaliana. So this is the plant. Okay, Arabidopsis thaliana. Here you see the importance of this plant. It's just like the Escherichia coli. Escherichia coli is a model organism, okay, for the genetic studies. For several studies, Escherichia coli or E. coli is utilized because it is the simplest, one of the simplest organisms. It is a bacterium. It is very easy to study this organism. So most of the scientists used E. coli as a model organism. Okay, and Mendel, he used Pisum sativum as a model plant for the genetic studies, Pisum sativum. And Morgan used Drosophila as an animal model for the genetic studies. Plant model is Pisum sativum used by Mendel, Gregor Mendel. And animal model to study the genetics is Drosophila by Morgan. And also the plant model, the simplest, one of the simple plants. It belongs to this Arabidopsis thaliana, belongs to the family Brassicaceae. It is also the old name for Brassicaceae is Cruciferae. We already discussed. Poaceae is the old name for the it is a Poaceae is the new name for the Graminae. Graminae is the old name. Poaceae is the new name. Like that. Cruciferae is the old name, and the new name for this family is Brassicaceae. So Arabidopsis thaliana is very important plant for the studies, for the genetic studies, for the developmental studies, and it is the first plant to be sequenced. The total chromosomes of all the chromosomes of the Arabidopsis thaliana are sequenced completely. This is the first plant to be sequenced. DNA sequence of this plant is totally sequenced. This is the first plant to be sequenced completely. So Arabidopsis thaliana is very important plant, but the question here is how many chromosomes are present in the Arabidopsis thaliana? And there are 5 so there are 5 pairs of the chromosomes are present in Arabidopsis thaliana. That is the right answer. In Arabidopsis thaliana, there are 5 pairs of chromosomes. But here, the question here is they asked 3 chromosomes, 9 chromosomes, 7 chromosomes, and 5 chromosomes. But it is not the exactly right answer. Right answer should be 10 chromosomes. That means 5 pairs. Instead of five pairs, they just gave 5 chromosomes. So the nearest answer out of the 4 options, the nearest answer is 5 chromosomes, but it should be the 5 pairs of the chromosomes or 10 chromosomes. Okay, so you should remember the number of chromosomes in the Arabidopsis, 5 pairs, and the number of chromosomes in Pisum sativum, 7 pairs. That means 14 chromosomes. Frequently they may ask these questions. And micro injection of DNA. So you know, micro injection of this DNA in DNA transfer, because in the genetic engineering or plant molecular biology, whatever you say, molecular biology experiments, usually the DNA of one organism is transferred to another organism. That study is called genetic engineering. So genes are transferred. Genes are genes in the form of the DNA is transferred from another organism to another organism. For example, to produce the transgenic plants, you know, BT cotton. BT cotton is produced by transferring the Bacillus thuringiensis gene. BT toxin. Bacillus thuringiensis is a bacteria. From the bacteria, that BT gene is transferred into the cotton plant. So that plant is called as BT cotton. But how that BT gene is transferred from Bacillus thuringiensis bacterium to cotton plant? How it is transferred? So the transfer of the genes in the form of the DNA from one organism to source organism to the target organism. How it takes place? By the some of the methods. The previous methods are indirect transfer. That means using the Agrobacterium tumefaciens. Agrobacterium tumefaciens is a bacteria. It has a plasmid, very famous plasmid, that is called as TI plasmid or tumor-inducing plasmid, TI plasmid. So this scientists use TI plasmid as a vector to transfer the BT toxin from Bacillus thuringiensis bacterium to BT cotton. So this is the indirect method. It is not directly transferred. One vector is used, Agrobacterium, the TA plasmid of Agrobacterium tumefaciens used. So it is called indirect method. Okay. So now you see here direct and indirect methods. Now I already discussed about Agrobacterium mediated gene transfer. The TA plasmid of the Agrobacterium is utilized. Okay, it was earlier utilized to transfer the BT gene into the cotton plant. So it is indirect method. And direct method, there are several direct methods. One is gene gun, biolistic method. It is also it is a gun. It's just like a gun. So what we do, they use the bullets or microprojectiles. They are also called as microprojectiles. Usually tungsten material is used, very small particles. It is also called as particle gun. Small particles are kept in the DNA solution, and those particles are shot into the plant or the callus of that plant in the tissue culture experiments. So along with these particles, that DNA is inserted into the tissue by using this gene gun or particle gun. So this is one of the methods. And micro injection, what is this micro injection? Just like injection, we know, we take the injection to our hands. Just like this DNA is injected. Okay, DNA is injected into the callus or embryos of that plant, target plant. So we can directly inject that DNA by using the micro injection method. Okay. So electroporation, one more method is called as electroporation. What we do, we keep the target cells. We can also mostly we use the protoplasts of those cells. Protoplast means the cells without the cell wall. Those are called protoplasts. So we keep the protoplast in the DNA solution and we apply some electricity, some impulses, electric impulses are applied. So then due to this electric impulses, this membrane becomes permeable. Permeable means it allows the entry of DNA from the outer solution to inside the protoplast. So these are the direct methods. That means directly without using the vector, DNA enters into the target plant tissue. So these are the direct methods. And indirect method is Agrobacterium mediated gene transfer. So you have to remember all these methods. So here the right answer, micro injection of DNA is a direct method. Okay, using cosmids, plasmids, and agrobacterium, TA plasmid of agrobacterium. These are all indirect methods, vectors. Okay, next. Uracil in RNA is chemically. So, uracil, there is, we already discussed DNA and RNA. There are some differences between the DNA and RNA. DNA is double stranded, whereas RNA is single stranded, right? And in the DNA, we see adenine, guanine, thymine, cytosine. These are the nitrogen bases present. Okay, we also call them as purines and pyrimidines. Adenine and guanine are the purines, and pyrimidines, thymine and cytosine. These are the purines present in the DNA. But the purine which is present in the RNA instead of thymine is uracil. Instead of thymine, instead of T, we find U, that is uracil in RNA. So what is this uracil chemically? Is it a carbohydrate? No. Is it a lipid? No. Is it a purine? No. Is it a pyrimidine? Yes. Because we already discussed that. So you see here. Here you see the structures of adenine, guanine, cytosine, uracil, and thymine. So these are adenine and guanine. These two are called as purines. Okay, they have two cycled structures. Okay, so and cytosine, uracil, and thymine. These are called as pyrimidines. Okay, monocyclic. These are monocyclic structures. And cytosine and thymine are present in DNA, and whereas uracil and cytosine are present in the RNA. Instead of thymine, uracil present in RNA. And instead of uracil, thymine is present in DNA. That you should remember. And these three are called as pyrimidine. So the right answer to the question is pyrimidine. Uracil is a pyrimidine. So you have to once again you go through what are the purines, pyrimidines, and how many. And adenine always pairs with T, that is AT. A and T have double hydrogen bonds. And GC, that means guanine and cytosine, they have three hydrogen bonds. Okay, so it pairs with always A pairs with T and G always pairs with C. That you should remember. There are three hydrogen bonds between G and C, two hydrogen bonds between A and T, and there is a Chargaff law. We'll once again later we'll discuss what is Chargaff law. Okay, so now the one more question is the steroidal lactones discovered in 1979 considered as plant growth hormones. So what are the plant growth hormones? Okay, these plant growth hormones are auxins, gibberellins, cytokinins, abscisic acid. All these are the phytohormones. Plant growth hormones also called as phytohormones. So you know about the auxins. What is the role of auxin and what is the role of gibberellin, cytokinins, and abscisic acid, ethylene. You should also remember ethylene. It is one of the most important questions because this is the only plant hormones in the form of the gas. Okay, ethylene, CH2, CH2. So it is the simplest, one of the simplest hormones, and in the gaseous. And here the question is about the steroidal lactones. These are the latest phytohormones discovered. These are salicylic acid, no. Jasmonates, no. And salicylic acid is a chemical. It is related to, okay, it is related to plants only. Okay, it induces the resistance in the plants. Okay, it is very important. Jasmonates also, these are the chemicals which are related to the pathogenesis. Okay, related to the resistance of the against the pathogens. And uh, strigolactone, it is not the right answer. And brassinosteroids is the right answer. Why they are called brassinosteroids? Because they are initially they are detected in the Brassicaceae family members. So they are like the steroid compounds. They are detected initially in Brassicaceae family plants. So they are named as brassinosteroids. And now about the brassinosteroids. You see, brassinosteroids are very important because they have several features. Due to the presence of the brassinosteroids, they give heat tolerance. In the plants, if there is more temperature in the summer season, particularly temperature increases drastically. So due to that, it produces some shock that is called as heat shock. It produces some heat shock proteins. Okay, so this brassinosteroids give the heat tolerance. Due to the presence of brassinosteroids, they can tolerate the high temperature also. Okay, cold tolerance. If there is very less temperature, also they can survive. That is called as cold tolerance, photoprotection. And metal sequestration. In presence of the heavy metals also they can grow. That is called as heavy metal tolerance. And organic pollutant tolerance. If there are organic pollutants also they can survive. And salinity tolerance. If the salt concentration is more in the soil, saline soil, also they can grow. And drought tolerance, they can grow when there is scarcity of water. Okay, so by ABA regulation, abscisic acid. So these are all the features of the brassinosteroids. Due to the presence of the brassinosteroids, plants show all these features. Okay, so that is the answer for this question. And next, brassinosteroids, you should remember, these are the steroidal hormones. Thomas Hunt Morgan, T.H. Morgan, propounded the theory of. So he proposed a theory. What is that theory? Chromosomal theory. Who proposed chromosomal theory? And law of segregation. Who proposed law of segregation? Linkage and crossing over. Who proposed linkage and crossing over? And law of dominance. Who proposed? Law of segregation, law of domination. These are dominance. These are proposed by Gregor Mendel. Mendel laws. Law of segregation, law of dominance. Okay, and here and chromosomal theory. Who proposed chromosomal theory? Sutton and Boveri proposed chromosomal theory. You have to remember who proposed chromosomal theory. Sutton and Boveri. And linkage and crossing over. Who explained linkage and crossing over? Linkage and crossing over. Where we find this linkage and crossing over? What is this linkage and crossing over? You have to remember. Okay, what is crossing over? The recombination between the homologous chromosomes during the packaging stage of meiosis 1. Crossing over, crossing over takes place between the homologous chromosomes during the packaging stage of meiosis 1. So that is about the crossing over. And linkage, what is linkage? Linkage means the genes which are present in the same chromosome, they are transferred from the parents to their daughter organ. Okay, their pedigree. So it is called as linkage. Okay, this was explained by T.H. Morgan. Linkage and crossing over was explained by T.H. Morgan. And the chromosomal theory was proposed by Sutton and Boveri. Law of segregation, law of dominance, law of independent assortment. One more thing. You see, Mendel laws, law of segregation, law of dominance, and law of independent assortment. These were proposed by Mendel. So you remember all this. Who is the father of genetics? That is the most it is important and easiest question. Gregor Mendel is the father of genetics. Okay, that we know. Charles Darwin is related to evolution theory. Okay, so it is not related. And Hugo de Vries, you know, he proposed mutations. Okay, uh, and the first formed product of C3 cycle. Now we came to again plant physiology, photosynthesis, where there are some cycles in the dark phase. C3 cycle, C4 cycle, and CAM cycle. Okay, why it is called C3 cycle? Who proposed C3 cycle? So there are different questions on this same topic. Okay, Calvin. Calvin, C3 cycle is also called as Calvin cycle. Okay, so Benson, all these are the different scientists. Particularly, you have to remember Calvin. C3 cycle is also called as Calvin cycle. Why it is called C3 cycle? So, what is the first product which is formed in the C3 cycle? The first product which is formed in the C3 cycle is 3-phosphoglyceric acid. It is also abbreviated as 3-PGA. And this 3-PGA has 3 carbons. It is a 3-carbon compound. Okay, it is a 3-carbon sugar, phosphoglyceric acid. It is a 3-carbon compound. So this cycle is called as C3 cycle. The first product of the C3 cycle is 3-PGA, phosphoglyceric acid. It has 3 carbons. Okay, so it is called as C3 cycle. And you know one thing, the in this reaction, in the first reaction, there is one famous enzyme is present that is called as Rubisco. Ribulose bisphosphate carboxylase. That is a full form of Rubisco. It is the most abundant protein present on the earth. This is one more important question. The most abundant protein present on the earth is Rubisco. Ribulose bisphosphate carboxylase. Okay, it catalyzes the first reaction in the C3 cycle. In the presence of the carbon dioxide, that reaction is called carboxylation, because it takes the carbon dioxide. In the first reaction, carbon dioxide is taken, so that is called as carboxylation. In the presence of the Rubisco, if the carbon dioxide concentration is more than 40 ppm, carboxylation takes place, and 3-PGA is the product form. But there is one more thing, photorespiration. Because Rubisco acts both as a carboxylase and oxygenase also. What is oxygenase? If the carbon dioxide concentration in the atmosphere is less than 40 ppm, then if the oxygen concentration is more, then it leads to another cycle that is called as photorespiration. Okay, so you have to remember this. Next, the primary carboxylating enzyme in C4 cycle. So now they asked, what is the first product in the C3 cycle is 3-PGA, and what is the first product in the C4 cycle? OAA, oxaloacetic acid. It is the 4-carbon compound. Okay, so here that is called as PEP carboxylase. That enzyme is called as PEP because the PEP, in presence of the carbon dioxide, it forms OAA. The first reaction is like that. PEP plus carbon dioxide gives rise to OAA. oxaloacetic acid. That is a 4-carbon compound. So that cycle is named as C4 cycle. Hatch and Slack. Who proposed this cycle? Hatch, Slack. And in India, particularly in our from the HCUs, Central University, Professor V.S. Ramdas, he worked on this. And he found this cycle in several other Amaranthus family members. Okay. So next, the question is the primary carboxylating enzyme in C4 cycle, carboxylation reaction. That means taking the carbon dioxide. That is the first reaction. The enzyme participates is called as PEP carboxylase, phosphoenol pyruvate carboxylase. Okay, that is the answer. And cohesion tension theory was proposed by who proposed cohesion tension theory? So there are ascent of water. Okay, ascent of ascent of sap. It is also called as ascent of sap. That means how the water against the gravity, how it reaches to the branches or the shoot tips from the roots. So in the ascent of sap, there are several theories proposed, and this cohesion tension theory, it was the most popular theory and proposed by Dixon and Jolly. And you know, our Indian scientist who got Nobel Prize, J.C. Bose, Jagadish Chandra Bose, who proposed pulsation theory. This is also explains the ascent of sap. Okay, but the theory proposed by J.C. Bose is pulsation theory, that you have to remember. Okay, and root pressure theory proposed by Stephen Hales. Root pressure theory. So there are several theories. They are also called as vital theories. Okay, root pressure theory, pump theory, water pump theory. Okay, these are Gard-Lewski proposed that. So these are all the different theories, and who proposed those theories you have to remember. And next, whiptail of crucifers is caused due to the deficiency of. So there are different deficiencies we can observe in the plants. So molybdenum deficiency, due to the molybdenum deficiency, we observe the whiptail of crucifers. Crucifers means brassicaceae family members. We find this whiptail, just like a tail, whiptail. We use for the horses. Okay, next. Stratification is useful in. What is stratification? So dormancy, we know seed dormancy. What is seed dormancy? The seeds are not able to germinate due to some factors. One of the factors is the thick seed coat. If the seed coat, outer seed coat is thick, then seeds take lot of time for germination. The inability of the seeds to germinate is called as, there are some external factors and internal factors also. That phenomenon is called as seed dormancy. So one of the methods to remove seed dormancy is stratification. That means rubbing the seeds by friction. Just by rubbing the seeds, it is a mechanical method. The thickness of the seed coat is reduced. Then they can germinate. And sometimes what they do, they burn the seeds. The seeds are very thick. Okay, then seed coat is very thick. Due to burning the seeds also, the thick coat is burned away. They can germinate. In the forest fires, this is what happened. Terminalia Arjuna, Tellamatti. It has very thick seed coat. They have to burn it literally. Otherwise, they won't germinate. So stratification is the method. Removal of cotyledons, no. Removal of embryo, no. Removal of dormancy due to hard seed coat. That is the right answer. Removal of dormancy due to hard seed coat. Seed coat should be, the thickness should be reduced by stratification method. And that is the right answer. The pigment helpful in the process of flowering is, that is called as phytochrome. Okay, and photoperiodism. This is also called as photoperiodism. That light influences the flowering process. Some of the plants flower during the winter. Some of the plants flower during the summer. This is the period of light period, daytime. It influences the flowering. The daytime is more in case of the summer, and duration of the daytime is less in case of the winter. So it influences the flowering. Though those phytochromes are responsible for this photoperiodism. So the pigment helpful in the process of flowering is phytochrome. That is the right answer. The movement of substance from an area of high concentration to low concentration is called as diffusion. That is the right answer. And next, the term enzyme was coined by who coined the term enzyme? Pune. He is the scientist. First term gave the term enzyme. And next, the stomatal arrangement where there are three unequal subsidiary cells surrounding the guard cell. So there are different types of the stomata in dicots: anemocytic, anisocytic, okay, parasitic, diacytic. These are the four types of the stomata we find in the dicots. Here, in case of the anisocytic stomata, we find there are three unequal sized subsidiary cells. Okay. In diacytic stomata, we find only two subsidiary cells surrounding the guard cells. In parasitic also, there are only two subsidiary cells surrounding the this one. So in anemocytic, there are more than three. There are more than three subsidiary cells in case of the animal. So